which normally you wouldn't really associate with 3D printing. But what we have is a platform of an externalized immune system, or the EXIS platform. I just talked about the traditional drug discovery that people do, which is you use mouse models, hybridomas, you use phage display, things that the pharmaceutical company has been doing for a very long time. And there are upsides to it and there are downsides to it. The biggest downside being it is a humanized system, which means it is animal derived that you then make it fit for human purpose. And what we are doing right now is actually sourcing these cells of interest directly from humans. So it's a human to human antibody development system. And what we're doing is engineering the organoids to essentially generate the kind of immune response you want, so that the B cells in question generate the antibodies we want. So that essentially makes it a very target agnostic platform for antibody generation. That means we can develop antibodies or therapeutics for cancer, inflammation, obesity, cardiometabolic, depending on what the target is and what the interest is. It is an agnostic platform. And the way we actually do that is via using 3D models and 3D organoid systems. We are recapitulating essential elements of the lymph node, which is where all of the antibody production of your body happens. And within the lymph node itself, there are these things called the germinal centers, which is essentially where all of the B cell activation and interaction with the T cells actually occurs. That gives you very high affinity, highly specific antibodies. And the reality of the system is, in our human body, this system is extremely sophisticated, very complicated, hard to actually replicate outside. But that is what we are trying to do using a 3D printed system. We have what we call the holographic two photon lithography printing, which is a two photon printing system that we use to 3D print our scaffolds that we then use as a platform for generating our organoids. So we have been doing that and have been, relatively successful. Like I said, there are elements of biomolecules, biomaterials, the form, and the cells that are involved in actually generating these organoids that we are then doing our drug discovery with. Again, a completely offshoot application of a bioprinted or 3D printed construct, which is completely relevant from a clinical perspective.